seguro editarlo.
Hola, mami. Thank you for joining us live today. I have a special guest, my husband, Dr. Manuel Lopez, who has published today some amazing work that he has been working on at UCLA in regards to autism. And I am so proud of him and his work and so happy that he agreed to come live here on Instagram Live and talk about his findings, which I know will help so many people, and that was his main goal when he started working. So let's start from the beginning. So I'm gonna put my glasses on because I see some people. Hi. If you have any questions, go ahead and comment below and he will be more than happy to answer them. So first of all, he's been working at UCLA for how many years now? 11 years on this amazing project. And can you tell us a little bit why you started this work and a little bit about the work itself?
Sure. Hi everyone. The reason because we started this work is because it was already known that some genes get the people predisposed to get some neuropsychiatric disorders such as autism. However, not all of them, they have the same condition. So this should be something else besides the mutation. So for the first time, we proved that there is a correlation, there is an interaction between some mutations and environmental factors such as infection. And we found the mechanism and the molecular basis responsible for autistic phenotypes. We found that early postnatal immune activation, we found that infections during the first years of life could be responsible for autistic phenotype. And we have all the mechanism responsible for that. We have different laboratories involved in this project with convergent results. We have human data. We analyze more than 3.5 million historical records. And our hypothesis actually is what is happening in humans. We found, and you can find it in the paper recently published today.
Link in my bio if you want to read it and share it. Great information. Go ahead.
You can find that we found in humans a prevalence between a relationship between the prevalence of infection during the first years of life and the development of ASD, from autism, in humans. So we found, if you have your kids between 1.5 years, so 18 months, and 4 years, you want to avoid them to have infections.
So from newborn, you mean from newborn?
18 months.
From 18 months.
From 18 months to 4 years, we found a correlation that those kids that they have an they have records, hospital records of infections. Mm-hmm. They have a really high correlation with the development of ASD. So we found that more than 40% of boys, not girls, boys, more than the 40% of boys who are suffering chronic infection or some kind of infection strong enough to go to the doctor, sometimes to be hospitalized, they develop ASD.
So I know that at this point, the data that was researched or looked at was from years ago, right? So the team went ahead and looked at all records, you said hospital records of kids or from 18 months old to 4 years old. That's what the records that were looked at?
Yes, we developed our research in a mouse model.
Okay.
So why? Because we was working with a mouse model corresponding to a condition in humans, is tuberous sclerosis, that they have more prevalence of ASD. So for having numbers clean.
Okay.
The prevalence of ASD, the prevalence of autism in the general population is between 1-2%. However, people with tuberous sclerosis, mm-hmm. they have a prevalence of ASD between the 40 to 61%.
Oh, wow, that's big.
And it's not the only genetic condition that predisposed to ASD. Okay. Fragile X, neurofibromatosis 1, all of them, they have a higher prevalence of ASD than the general population.
Oh, wow.
So that's why we work with this mouse model. Because if we want to study autism, we have to work with the model that has autism.
Right.
So we were able to trigger autistic phenotypes on those mice, mimicking viral infections. And we were able to reverse and prevent this phenotype.
Wow.
Being able to prevent and rescue it even when they are adults.
So let me explain what that means. Prevent ASD in these mice. It's amazing. And rescue it means curing it, basically, right?
In mice, yes.
In mice, right?
In mice.
But
In mice, we were able to trigger autistic phenotypes, and we were to cure, to reverse these autistic phenotypes.
But what you use to cure it or prevent it or reverse it, can you tell us about that drug?
Sure. We found, as I mentioned, we have the mechanism underlying of this phenotype, of this condition, why these mice developed ASD. So we found the cellular type responsible, that is microglia. We found the molecules responsible, that is interferon. That interferon is what the body produces when you have viral infections. So be careful now with the COVID around.
I was going to get to that. Like now there's COVID.
And in case of TSC2, we found that the TSC2 mice, they have higher level of mTOR. mTOR is a growth factor that everybody has. But the patient with TSC2, the mutation, the tuberous sclerosis, or our mouse model, they have higher mTOR. So we use rapamycin to prevent these abnormal elevated mTOR levels, and we rescue the phenotype.
And
forever.
rapamycin is the drug.
And rapamycin is the name of the FDA drug approved.
ADA approved drug. So
FDA,
sorry. I said ADA. So this FDA approved drug, does this mean that in the near future it could be tested to see if
I don't see why not.
So to see maybe if humans are if it's able to work just like with. So first of all, why on mice? Why do you do your research on mice? Like,
well,
how do they compare to humans? Like, what does that matter?
It is relatively easy to work with a mouse model that they have a mutation that we carry on humans. So these mice, they are transgenic mice, that they are just mice that they are carrying on one of the mutations that in humans are they responsible for the tuberous sclerosis condition.
Okay, and how related are they to the way that humans are? Like,
well, mice and human are not the same, obviously. But they're closer in the genome more than we think. Actually, you and I, we are not even so close in our genome. So the people around, and actually the study that we present in this manuscript, the human data is not people with tuberous sclerosis, it's general population.
Oh.
Because we found the mechanism responsible for the autistic phenotypes that we develop in our mice.
Okay.
And we rescue this phenotype. When you know the mechanism, you can just target specific points and you can just rescue it.
That's amazing.
And we did it. So once we develop our model, we contact Dr. Andrea Reski in Chicago, and we ask him. He's a statistics, a statistician, and he has access to millions of data, human data. This is what they do, analyzing data.
Oh, wow.
So we contact them and say, hey, we have found this in mice. Can you check if this is happening in human? Because our prediction is like if you have immunoactivation in the first years of life, you have a high chance to develop ASD. Okay. So he did the analysis, and actually, he when we contact him after he did this analysis, he told us that he found the higher correlation that he found ever between the immunoactivation during the first years of life and the development of ASD, and not only ASD. This work has been more focused on ASD, but it is also related with other neuropsychiatric disorders. And not all these people involved in this study were not just tuberous sclerosis people.
Okay.
They were general population, because again, the genome of each of us, they have plenty of mutations that they're silenced. And silence means that by themselves, they don't give you a phenotype. They don't give you any problem by itself. But when in combination with environmental factors such a viral infection in this case, or could be the contamination, smoke, all of these can be the turning on the problem because of these mutations.
Wow, that's impressive. So it was general population. That's even more impressive. You said, what percentage did you say it was in children and boys?
The odds to get the ASD is with a very strong statistical significance is more than 40%.
That is amazing. This is shocking news. And this is amazing that you and the team of collaborators were able to, connect the dots, and I'm so proud of you.
Remember thank you. Remember the autism, the prevalence of autism in general population by themselves is 1 to 2%.
Wow.
And we found that in combination with immune activation, specifically in this time window between 18 months and 4 years, you go from 1% to 40% or above.
Okay, what does that mean for, like, right now we're, so COVID, it's a huge thing right now, like the last 2 years. So parents have to be extra careful, for their
Parent
kids.
has to be extra careful, and parent has to be smart.
Okay.
And when I say smart, and I'm going to say it, is vaccinate your kids.
Right, because your research has actually found exactly that there
I
is
know,
a correlation.
exactly. I know it's kind of controversial. Some people don't want to talk about it. Some don't want to hear about it. But please, this paper is a keystone of evidence. It's a accumulation of results from different laboratories, from different mice model, from different human model, that you can show that here is. If any of you hear this and think, oh, immune activation, that's vaccine. They say, no, that's a big no. That's a big no. Let me put you this in perspective and with easy words. If you don't get your kids vaccinated for polio or for flu, they can get the flu or they can get the polio. And if they don't die because of the polio or the flu, they can be exposed to the high immunoactivation when they are kids. What it means, like, they have a more than 40% of chance to develop ASD.
In boys.
In boys.
Okay.
It's already known that the autism is more prevalent in boys than in girls. It's like 4 to 1.
Oh, wow.
So actually, in our mouse model, a male developed the phenotype, but not the female. But in this work that I recommend you to read it, we found why. Because in addition, we found what is the cell type responsible not only for this phenotype, but why the male are more vulnerable than the female.
Why?
Because of the microglia. The microglia is a cell type that we have in the brain that now it's getting more popular, but for a long time has been considered just a helper in the brain.
Okay.
It's just structural, it's just for cleaning, it's this kind of thing. But no, we found and we describe in this paper that it has a critical role. So historically, if you talk with a neuroscientist and you ask what is the responsible for, they will start the answer with the neurons.
The microglia is responsible for neurons.
But historically, every neuroscientist, or the 99.9, they answer the neurons are the responsible for.
Yeah, okay.
And it's not, even if the neurons are involved, of course, it's one of the main cell type in the brain.
Okay.
It seems that the neurons are, at least in these conditions, the neurons are doing what the microglia says them to do.
Whoa, that's like huge.
Yes.
Wow. So this is how many years of work again? Can you please share with everybody so they know that
11 years.
11 years of work and research, and also all of your experience from before. Before that, he's done work with, I want to say Alzheimer's, but it's just, what do you call it?
With what?
Alzheimer's, your other science paper?
Yeah, it's about memory.
It's about
memory. Learning and memory.
So this is coming...
Because neuropsychiatric disorders, all the deficit in cognition, they are going with the learning and memory deficit.
Right.
So if you go to the mechanism underlying that, this is what we try to do here, is just fix the problem. It's just fixing the problem. Try to do research to present a problem. It's relatively easy and relatively short. So we found that we can trigger autistic phenotypes in our minds like 6 years ago, 7 years ago.
Oh, wow.
That's kind of relatively easy. But the thing is, like, we don't want to present to the public something that say, hey, immune activation induces something autistic phenotype. Because we don't want to bring more noise to this kind of controversy about vaccination.
Okay.
That it shouldn't be controversy. Please vaccinate your kids.
So you brought proof. You took even
show a problem.
6 more years to make sure you fixed the problem, and you're able to help families in this way. So
show that there is a problem. From the science perspective, it is cool because it's one more step for understanding what's going on. But from the human perspective, if you don't have a solution for that problem, what's the point to scare people?
Right. I like that. That shows a lot of ethic from your part, because somebody that just wants to publish right away, whatever, like that's
no. We need to be sure that what we present is solid enough and conclusive enough that nobody can actually doubt about it. And it's not doubt about it, the result. The result has been replicated already in our lab, in different labs, to be sure that this is what it is.
That's the peer review aspect of it.
Yeah.
Right?
The point was, it's just these results support the idea, emphasize the idea, beg for the idea that you need to vaccinate your kids. Because if you are not getting your kids vaccinated and thinking that you are protecting them from autism, this work, again, I repeat, replicated with convergent results from different laboratories with mice and human data, supports the idea that you are doing it for the opposite. If you don't get your kid vaccinated, they have higher chance to get immune activation. What it means, they have a higher chance to get ASD.
Wow, this is breakthrough. So, thank you.
You're welcome.
And at this point, I'm gonna see if there are any questions. I didn't announce this. It was just kind of like, let's do this, because today in particular, the article,
just today.
It was published today, so we're very excited that arduous work. And I know you're working on another really, really interesting and exciting paper, but this is breakthrough. This is news. This is something that everybody should be sharing and talking about, because everybody needs to know this. And I wanted to emphasize on the fact that this is somebody that takes his work very seriously, that has been working very, very hard and doesn't cut any corners. So, I mean, the credibility, all of his work speaks for himself. Being published in prestigious journals, and having the option when you graduated from your PhD, you had the option to go work at Harvard, go work at UCLA. Like, he had options because of his work. His work speaks for himself. So I just wanted to point that out. I'm so proud of you. And if you want to read the article, there's a link on my bio. Feel free to share it, download it, read it. Great stuff there. If you have any questions, go ahead and comment below. At this time, I'm going to